Hello everyone. Welcome to today's IVIM webinar. I'm Morgan from IVIM Technology and I'm very happy you could join us today. This webinar will highlight how advanced in vivo imaging technologies are transforming skin disease research and drug discovery with a particular focus on the real-time visualization in living tissues. Today, you will discover how intravital microscopy enables researchers to directly observe cellular dynamics within skin disease models, providing deeper insights into disease mechanism and the therapeutic responses. Through application, we will also explore how these approaches can enhance preclinical evaluation and accelerate the development of effective treatments. Next, we are pleased to welcome Professor Pirang Kim of KAIST Graduate School of Medical Science and Engineering and CEO of IVIM Technology. In this session titled Accelerating Skin Disease Drug Discovery Through In Vivo Imaging, he will demonstrate how intravital imaging can be applied to skin disease research, including atopic dermatitis, and how real-time in vivo imaging can support more precise and efficient drug development. And please don't hesitate to submit your question in the Q&A box at any time during the presentation. We will do our best to answer as many as possible during the Q&A session at the end. Additionally, if you're interested in assessing the latest data presented today, IVIM Technology's CRO services are available to support your research. You can find more information, including the brochure, in the resource tab below. So we encourage you to take a look if it's relevant to your work. Professor Kim, please begin.
Okay. Thank you. So thank you everyone for joining today's webinar. So let me begin. So today I will introduce this all-in-one real-time intravital microscopy solutions provided by IVIM Technology. And then I will also show you several examples of in vivo 3D visualization of skin in cellular level for skin research. Okay, so I guess, some of you may already heard about intravital microscopy. So intravital microscopy is a technique that can provide this microscale, cellular level imaging and video acquisition in a living tissue in a live mouse model. So compared to other imaging imaging techniques such as X-ray, CT, MRI, ultrasound, intravital microscopy can provide real cellular level resolution imaging in a live mouse model. So this particular video was obtained from the lung of a live anesthetized mouse model. So in this example, we are visualizing the two things. The first, we are looking at the blood flow by using intravenously injected hepatitis dextran. So basically, we are doing fluorescence angiography here to see the blood flow. And at the same time, we are looking at the neutrophil in red color. So all this red dot is the neutrophil inside the vessel. And then these neutrophils were presently labeled by injecting antibody fluorophore conjugate, anti-LY6G antibody conjugated with the red fluorophore. And this antibody fluorophore conjugate was intravenously injected through the tail vein to label the neutrophil, as you can see in this video. And then we injected those antibody conjugated at 1 hour before this imaging. So we can nicely, label all the neutrophil inside the vessel. And then we can directly see their interactions and dynamics inside the vessel in the lung. So as you can see, we can see these neutrophils are interacting with each other. So they are, kind of connecting to each other and then make a cluster here. And then it physically block the vessel at these locations. And by doing that, it disturbs the microcirculation in the lung. Okay, so this is one of the very basic, pathophysiological mechanism happened in the acute lung injury model. And next, in addition to cell, we can also directly image the drugs, fluorescently labeled drugs in the live mouse model. So this particular example were obtained from the liver after the intravenous injection of a fluorescently labeled nanoparticle. So this is a real-time video, and that's why we can see this kind of a flickering. Those are the tissue motion induced by the mouse breathing. And in this example, we are looking at the blood vessel expressed. It's visualized by the using transgenic mouse model tied to GFP. So all this green color is the liver sinusoid. And then the red color is the fluorescently labeled nanoparticle injected through the tail vein. And then in the beginning, you can see how this nanoparticle is delivered through this liver sinusoid vessel, and then how they are accumulated inside the liver sinusoid, as you can see in here. So in addition to nanoparticle, we have been imaged antibody drugs, peptide drugs, exosome drugs, and other, drugs modality in vivo in a live mouse model. So in short, intravital microscopy is a quite useful technique to acquire a live cellular level images and videos in a live in vivo animal model with molecular and functional informations. Okay, so to do this intravital microscopy, IVIM technology is providing this neural mode intravital confocal two-photon microscopy system, the CMS3. And as you can see in here, this is an all-in-one packaged system with quite compact size. Okay, so it can be easily placed anywhere in the lab. And then this is a very much optimized system for in vivo, cellular level intravital imaging of various internal organ of a live animal model, as you saw in the previous slides. And then this microscope can do both of two-photon, four-color two-photon imaging and four-color confocal imaging. So it can easily switch over between these two imaging mode, two-photon microscope mode and confocal microscope mode. And then in addition to that, if needed, if necessary, we can also connect other additional lasers through the optical port behind the box, such as tunable titanium sapphire femtosecond pulse laser system. And then this microscope is equipped with very high-speed laser scanner for real-time imaging with a speed of 50 frames per second by 512 by 512 pixels per frames. And then based on this real-time imaging capability, we built in the automatic live tissue motion compensation function, which is very critical to obtain high-quality images and videos in a live animal model. And then this is how we prepare the mouse model for imaging. So this front door can be opened, and then we can connect the anesthetization system, gas anesthetization system, or we can use injectable anesthesia. But anyway, we can put the mouse model, as you can see, on the heated motorized stage. And then inside, this whole space is actually heated. So we can keep the mouse body temperature to be stable during the long-term imaging. And then we also have a built-in CCD camera to monitor the animal anesthesia level during the imagings. Okay, let me show you, several examples of four-color two-photon imaging of live tissue. To give you a little bit of a background, two-photon microscopy is a technique that can generate fluorescence signal only at the focus, as you can see in this photo. And then by using a longer wavelength near-infrared laser, it can penetrate the tissue more deeply. So by using longer wavelengths laser, it can penetrate the tissue better. By doing that, we can achieve deeper imaging in the live tissue. In addition to that, the two-photon microscopy is capable of label-free second harmonic generation imaging. So without any process labeling, two-photon microscope is capable of second harmonic generation imaging, which is quite useful to detect, to visualize the fibrotic collagen in a live tissue in vivo. So as you can see here, this second harmonic generation imaging has been used to visualize the collagen fiber or other regular protein fibers in a live tissue. And then this is one example. So this image was obtained from the liver of a live mouse model of liver fibrosis and steatosis. And then this green color is the fibrotic collagen accumulated in the liver, visualized by the two-photon microscope with the secondary generation imaging. And then at the same time, the fluorescence signal can be simultaneously detected. So we label the lipid droplet inside the hepatocyte using the ProFu SF44. So it can do both of the fluorescence and secondary generation imaging simultaneously to visualize the lipid droplet and fibrotic collagens. And it works beautifully, as you can see in this example. Okay, and then also in the skin, this secondary generation imaging is quite useful to visualize the dermal collagen. So we can nicely see the where the dermis is located in our imaging. So again, this is a four-color imaging data obtained from the skin of live mouse model, live anesthetized mouse model. So this is a G-stack video starting from the epidermis through the dermis. And then the green color is actually nuclei. It's visualized by H2B-GFP. And then the red color is the ROSA26 membrane tititometer. So we can see that every cell's membrane is red colors. And then we inject some other dye to the tail vein intravenously, so we can visualize the blood vessel, blood flow in blue color. And then finally, we are visualizing the dermal collagen by detecting this secondary generation signal. And as you can see in this single slice image, we can nicely visualize the every cell's nuclei in cellular level, in subcellular level. And then we can see nicely see the hair follicle. And then we can also nicely see these dermal collagens. Okay. And then this is a more maximum intensity projection image to show both of the hair follicle and the large vessels. Okay. And then as I told you, this microscope, our intravital microscope can be applied to other tissue as well. So this is one example obtained from the muscle. So again, this is a live tissue. And then in the muscle, the secondary generation signal is also strongly generated from the myosin fiber, myosin filament. So you can nicely see this myosin structures in white color, and then nuclei, and then myocyte. And then and of course, by taking multiple images at different location, we can generate this large area, mosaic image with automatic stitching functions. And then we can also visualize other internal organs such as pancreas. And then you can nicely see this actinide cell. This is the wide area images, in vivo images obtained from the pancreas with the motion compensations. And then this is the kidney, same thing. So nuclei and then cell membrane. And then we can see the tubule of the kidney. This is the large area images. Okay, so I just showed several examples, but our intravital microscopy system has been able to visualize more than 30 different tissue and organs in a live anesthetic mouse model. So all these images were obtained from the live mouse model. And then we have been able to image the brain, brain tumor, of course skin and hair follicle, and various tumor model. This particular example is a colorectal tumor model, pancreas, spleen, GI tract, adipose tissue throughout the body, prostate, placenta, lymph node, memory tissue, cell transplant, kidney, muscle, liver, heart, lung, and so on. And then I like to emphasize again, all these images were obtained from the live tissue in a live mouse model for through in vivo imaging. Okay, and then let me introduce about the real-time imaging capability. So as I briefly mentioned, when you image the live tissue in a live anesthetic mouse model, one of the biggest challenges is the tissue motion in microscopic scale. So, as you can easily imagine, even though we anesthetize the mouse model, still the mouse is breathing air. and then heart is beating, and the muscle is sometimes, moving. So all these physiological motion induce, this kind of, tissue motion in the internal organs. So, our approach is quite straightforward. So we just record the real-time video with fast speed enough to track all this tissue motion. And then we select the in-focus frame automatically and then do the image registration to generate these motion compensated images. So you need two things. First, you need the high-speed microscope, fast enough to track the tissue motion. And then you need a motion compensation algorithm to select the in-focus frames and then do the image registration in sub-pixel resolution, sub-pixel precision. And then this is the result. So yeah, you can see from this moving tissue, we can nicely generate this high quality, high resolution images. And then the typical imaging resolution of our microscope is 0.5 micrometer in the live tissue, in a moving live tissue in a live mouse model. And then without this motion compensation functions, you will, end up these very blurry images, as you can see in this example. And then this function is built into our software, and then this is automatic functions. So all you need to do is just turn on the motion compensation function and press the single button. And then in just a couple of seconds, it automatically generates this high quality motion compensated image inside you in a live mouse model. Okay, in addition to that, well, we can also, include, denoising, AI denoising algorithm. So this is our collaborative work with another professor, Young-Gyu Yoon at KAIST. And this is the real-time, denoising algorithm based on blind spot denoising algorithm. And then the good thing of this algorithm is it doesn't need pre-training, and then it's unsupervised method, which means this algorithm can be done automatically. And then this is very much optimized system, optimized the denoising model for high-speed microscopic video. So let me directly show the one example. So this is the real-time video obtained from the skin of live anesthetized mouse model. This is a low video. And then this is the high-speed video, high-speed real-time video. And then again, the green color is the nuclei, red color is the cell membrane, and blue color is the red blood cell, fluorescently labeled red blood cell, flowing in the capillary. And then after this automatic denoising, you can see the noise was very nicely removed, and then the signal to noise ratio is improved more than 100 times. And then we can nicely see this in all these subcellular level structures in very high-speed videos. So more tricky example. So this is more noisier. This video was obtained from the kidney, and then you can as you can see in this example, we can nicely remove the noise from this real-time video and generate this high-quality, high SNR real-time videos. And this one is more extreme cases. So the noise is very high, the signal level is very low. But anyway, this is the real-time video obtained from the brain. And then even from this noise video, we can, remove all the noise and then, make the unseen structures can be visible, as you can see in this area. Okay. So this AI denoising model works beautifully. And then we made this AI denoising denoiser software. This is automatic denoising software. So all you need to do is just one single button click, and then it automatically do all the thing and then generate the denoise real-time video. So not just although this software is optimized with our microscope, but other real-time video can also be denoised by using this software. So if you're interested, please send us an email to request the demo version of this software. So from now on, let me show you several examples of how we use our intravital microscopy for skin research. Okay, so let me start with one thing. So this example, in this work, we use the intravital microscopy to see the immune cell trafficking in the ear skin. So this is how we prepare the mouse model to see the ear skin. So mouse is anesthetized, And then we inserted this rectal probe through the anus to monitor the body temperature. And then we feedback control our heating pad to keep the mouse body temperature to be stable. Okay, and then the ear skin is attached to the glass here. And then this is the time-lapse video showing the neutrophil and macrophage movement in the skin. So you can see this cell movement. And at the same time, we label the blood vessel by injecting the anti-CD31 antibody conjugate with the fluoride fluorophore. And then again, this antibody was intravenously injected at 1 hour before the imaging, and it nicely labeled all the endocellular cell, so we can nicely see the blood vessel, as you can see in this example. Okay, and then this is the real work. So we've prepared one protein, which is called KLS, and then we labeled it with the fluoride fluorophore, and then we intradermally injected this protein into the dummies, as you can see here. And then we take a real time, the time-lapse videos at 3 hours after and 6 hours after. And then we can nicely see the recruitment of this neutrophil and macrophage to this injection site, as you can see here. Okay. So this is one example. So we can nicely see the immune cell dynamics in the skin by using the intravital microscope. In addition to that, we have done some other work. So we induced the psoriasis by using imiquimod cream, and then we can repeat the imaging in the single mouse model. So basically, this is just one single mouse model, and then repeatedly imaged at the day 1, day 0, day 1, and day 2. And then this is a collaboration work with another researcher at Kumamoto University in Japan. And then here, again, we detect the collagen signal by using second-order generation signal. So we can see where is the dummies. And then we label the blood vessel using CD31 antibody fluorophore conjugate, intravenously injected antibody. And then we can also image the neutrophil. And then if you compare this sham control, we can see the huge infiltration of this neutrophil at day 1 and day 2. And then, so let's compare the data here. So as you can see, we can nicely see the increased infiltration of the neutrophil. And then later, we find out this neutrophil were many of those neutrophils were originated from the bone marrow. Okay, so compared to sham, we can nicely see this neutrophil. And then this is the G-stack data. So again, so we can see many these red dots, those are the neutrophil infiltrated into the skin. So like this. Okay. And then as I showed you in the previous slides, the big advantage of the intravital microscope is we can save the mouse after the imaging. So we don't need to sacrifice the mouse after the imaging, which means we can repeatedly image the same single mouse model at different time point and see the sequential and the longitudinal changes over several days to several weeks, and which can give us more precise and more insightful data. Okay, and then also we can use intravital microscopy to monitor the drug penetration into the skin. So this is another collaboration work published in this journal. And then, so we made this so our collaborator made this nanoparticle, BRMP, and then they made their, atopic dermatitis mouse model, and then applied this nanoparticle on the surface of the skin. And then here, again, by looking at the collagen signal, we can nicely visualize the where is the dermis in blue color. And then we apply the BRMP on the skin surface. And then over time, you can see this BRMP is delivered, it's reached to the dermis, mainly through the hair follicle, hair shaft. And this is a little bit more later time point. This is the beginning, right after the applications, 6 hours and 12 hours after the application of BRMP onto the skin surface. And then, we can nicely see, we can clearly see the delivery of this nanoparticle through the hair follicle, hair shaft, and then some other channels. And then it and then it reached into the dermis. Those are, visualized by detecting the collagen signal, second amino signal from normal collagen, as you can see in here. Okay, another example. So another drug penetration imaging applications. So again, our collaborator made this peptide delivering nanoparticle. And then this is the result. So this peptide is labeled with green profile PTC. And this is the naked peptide. And then this is a nanoparticle packaged peptide. And then as you can see in here, we can nicely see the enhanced delivery of this peptide, packaged peptide nanoparticle into the reached dermis, as you can see in this example. And then we can quantify, how much penetration, skin penetration is improved by this type of packaging, as you can see in here. Okay, and then also for other skin disease, well, not disease, more exactly. But anyway, so in here, our collaborator prepared this functionalized microstructured film, and then this film, helped to accelerate the skin wound healing. So compared to here, so this is in vivo data. And then this white color is the second generation signal from the dermal collagen. And then we can see their, accelerated skin wound healing with this film. Okay, so in addition to this example, we can also see the other, cellular structure in the skin by using antibody labeling or transgenic mouse model. So on the left-hand side, this is the one. So all these images were obtained in vivo. And then we have been able to visualize the Langerhans cells, dermal discs, or dermal macrophages, as you can see in this example. And then this is an lymphatic vessel in the skin. And then we can also visualize the peripheral nerve in the skin. This other example. So as I already showed you, we can visualize the macrophage and neutrophil together with the vessel. And then we can also see the nerve in the skin together with the vessel. And then this example I already explained. We can visualize the neutrophil, vessel, and dermal collagen. So 3-color imaging in this example. Okay, so this kind of other cellular structures or skin resident cell can be imaged by using intravital microscope. Okay, so from now on, let me just briefly show you, the application on other organs, just briefly. So this is as I showed you in this slide, we have been able to image all other internal organs such as lung, or pancreas, and other organs. So to achieve the stabilized imaging of actively moving organs such as lung, we make this stabilized window. So so we suck out the air through this suction hole, and then we can achieve the very gentle negative air pressure inside this imaging chamber. So we need only -3 to -5%, negative air pressures. And then that was enough to stabilize the lung surface, as you can see in here. And then this air pressure, negative air pressure is very gentle, so we can keep the blood flow to be unimpaired, as you can see in this video. And after that, we applied automatic motion compensation to generate this motion compensated timeless video, which nicely shows the neutrophil clustering in the lung, as you can see in this data. And then just for fun, I like to show you one short video how we do this in vivo lung imaging. So we first need to intubate, connect it into the mechanical ventilator, and do the small surgery to expose the lung. And then we use our stabilization imaging window for the lung surface stabilizations. And then we put it into our microscope, and then the temperature heated and maintained. And then we can obtain, and then we focus into the objective lens, and then we can obtain the images. And also, I'd like to show you one example how we can, test the drug efficacy in this model. So this red color is the red blood cell. So it's visualized, basically we are visualizing the microcirculation. And then as you can see, this accumulation of neutrophil disturbs this microcirculation. So no red blood cell perfusion here. But after some drug treatment, so this is the before treatment and this is the after treatment. So with some drug treatment, we can directly see the recovery of the red blood cell microcirculation, as you can see in this example. So we identify the neutrophil as the cause of this microcirculation disturbance, and then by targeting this neutrophil, we can recover the microcirculation. So this is one good example how we can use intravital microscopy, intravital imaging for drug efficacy analysis in vivo mouse model. And then, as I showed you, as I told you before, we can image other drug modality. So in here, we labeled the exosome, and then this before injection, 10 minutes and 30 minutes after the injection. And then we can see that this intravenously injected exosome can specifically target the cell within 10 minutes, and then later it was internalized, as you can see here, at 30 minutes. All right, and then there is other, I like to briefly show you the imaging window technique. So first, dosage pole chamber. This is very well established window technique for cancer xenographic longitudinal imaging. So as you can see in this portals, we can permanently implant this dorsal skin pole chamber, and then we can inject the human cancer cell in the middle of this imaging window. As you can see in here, and then we fix and then we seal this space, subcutaneous tissue with a cover glass with a sealing. And after that, we can keep the mouse to 3 to 4 weeks, and then we can repeat the imaging. So like this. So by combining our intravital microscope imaging with whole body imaging systems such as IVIS, we can see both of macroscale and microscale images from the single mouse model. And then let me show you one example how we use this dosage pole chamber to monitor the drug delivery. So our collaborator made this nanoparticle that can specifically target the tumor activated endothelium. So we made this dosage pole chamber mouse model with triple negative breast cancer cell lines, MDAMB231 GFP. So after injecting this MDAMB231 cell into the dosage pole chamber, we wait 2 to 3 weeks to this tumor cell to grow. And then we label the blood vessel again with the CD31 antibody, and we inject this nanoparticle through the tail vein. And this is 2 hours, 6 hours, and then 24 hours after the injection. So we can repeat this imaging at different time point. And then we can find the exact same location inside this imaging chamber to do the longitudinal imagings. And let me zoom in. So this is how this nanoparticle is delivered. And then if we zoom in, you can see that this increased the delivery of the nanoparticle over time from 2 hours to 24 hours. And then we can finally see the uptake of this nanoparticle to the cancer cell. So the beauty of using this imaging window is we can find the same location, exactly same location at different time point for long-term longitudinal imagings. And this is another example. This is the memory imaging window. So we can see the orthotopic breast tumors. And then we can do the similar type of imaging analysis with the memory tissue. And then let me briefly show you the abdominal imaging window technique, which is a ring-shaped window that can be implanted onto the abdomen or side of the mouse model to visualize the internal abdominal organs such as liver, kidney, pancreas, and spleen. So this is our preparation, the implantation of this imaging window to the side to visualize the kidney. And then, again, as I told you, the advantage of using this imaging window is we can find the exact same location at different time point. So we induce acute kidney injury, and then we find the exact same area, and then we repeat the imaging from day 0, day 1, 3, 5, and 7. And we can see how this single tubule changes over time and how individual cells are reacting, changes over this 1 week time period. So we can nicely analyze this disease progression in cellular level by using this abdominal imaging window. And then we can also apply to the pancreas as well. So again, in here, we make an orthotopic pancreatic tumor model by using this PANCO2 GFP cell. So we inject the pancreatic cancer cell here, and then we can keep the mouse to 2 to 3 weeks. And then we can directly see how this tumor cell grows and infiltrate into the neighboring tissue like this from day 11 to day 18. And then we can also visualize how this tumor vessel changes over time. And then we can decide, and then we can do the drug delivery monitoring and then drug efficacy monitoring in the pancreas by using these techniques. And then let me finally cranial imaging window technique, which is also very well established technique for long-term repeated brain imaging. So this is the procedure. So we make a small hole with a 3 millimeter diameter hole in the skull. And then we put the transparent cover glass on top of it, and then protect the skull with a dental cement. And after this surgery, this mouse can survive more than 1 year. So we can do very long-term longitudinal brain imaging. And then when you do this surgery, we can keep the dura matter intact. So we can see the dura, meninges, and brain cortex using our intravital microscope. And this is the cellular level visualization, starting from the dura and the meninges and pia mater and brain cortex. In this particular example, we use NG2 reporter mouse, so we can see the smooth muscle cell and capillary parasite in the cortex. And then also, by using this cranial imaging window, we can do drug delivery, imagings in the brain. So as you can see, this is a 48 hours, longitudinal imaging. And then this time we monitor the antibody drugs. So this red color is the antibody drugs. Upper panel is unmodified version, so control antibody drugs. And lower panel is modified antibody drugs to make it can be delivered across the blood-brain barrier. So as you can see, these control antibody drugs, they cannot penetrate the BBB, so they just remain in the vessel, remain inside the vessel, and disappear over time. But after this modification, we can see the increased delivery of these antibody drugs to the brain parenchyma, as you can see here at 24 hours and 48 hours. Okay, this is one single mouse model, and then we can directly see this, drug delivery, antibody drug delivery in the brain. And then another study we have been done is we can easily identify the penetrating arteriole, as you can see in here, through the cranial imaging window. And then by using photoreactive chemical Roche Wenger, we can make a thrombus in the brain. So inside this penetrating arteriole, we can make the photothrombosis. So, and then the flow was disappeared. And after that, then we can find the exact same location in the brain from day 0 to day 30. So this is, again, one single mouse model, longitudinal imaging, and then we can see the cellular level changes over time. So this is another data. So this is the same vessel. This is the exact same vessel, so we can find the exact same locations. And then, so we can see how individual cells changes from day 0 to day 1. So this cell change like this cell change like this. Okay. And then at any time point, we can also do the time-lapse imaging and then see how the cell and the cell dynamics, as you can see in this example. Okay, so for all these different organs, we are providing this imaging window solutions, and then that is all these imaging window kit is optimized with our microscope, and it's ready to use. Okay, so in addition to our standard model, we are also providing the other model, FS model, which has a more large open space. So it can easily handle the large animal model, medium-sized animal model such as rat, parrot, and then even rabbit. And then, of course, from the lab, we can also obtain these cellular-level images from all these different organs. And then for larger animal model, we have another version, which is called Roboderm integrated microscopy system. So we miniaturize our microscope, and then everything is delivered through the optical fiber. And then this Roboderm is a human cooperating version. So we can put our microscope anywhere in the large animal, such as pig or even non-human primate. And after we place our microscope, then this robotic arm can be controlled in micrometer scale, micrometer precision. And then we can obtain these cellular-level images. Okay. And then although our microscope is designed for the more optimized for the live animal imaging, it can also image the standard slice sample like this. And then we also providing the our dedicated intravital image processing and visualization software, IVIM Studio. And then by using it, we can nicely deal with this large area 2D imaging data, and then 3D data such as long organoid model. And then we can do all this analysis of JPEG images, and then 3D rendering, and then section imaging, and then visualizations. And then this is the 4D, data, videos obtained from the skin. Okay, and then we can nicely see how these individual cells are moving around inside the 3D skin space. Okay, and our microscope has been installed. It's installed all these different universities and hospitals and companies worldwide. Okay, so this is my last slide. So if you're interested in this technique, please visit our webpage, www.ivimtech.com, or just send us an email to information@ivimtech.com. Thank you very much for your attention.
Thank you, Professor Kim, for the excellent presentation. We'll now open our Q&A session and wrap up today's webinar. So there's several questions.
Okay, so all right, in Q&A, okay, so I can see the first question is, has it been confirmed whether BRMP delivered to the dummies is actually taken by target cells like immune cells or fibroblasts? Yes, we can. So we can visualize the specific immune cell in the skin or fibroblast, as I showed you in the examples. So if we can label the BRMPs, any drug molecules, then we can clearly see whether those drugs or whatever molecule is uptaken by the target cells. So short answer is yes. All right, and the next question is, in the imiq mode-induced psoriasis model, did tissue inflammation during repeated imaging from day 0 to day 2 have any impact on imaging quality? Yes, indeed. Actually, so with the psoriasis imiq mode-induced psoriasis model, as you know, the epidermis gets thickened over time. And then due to those, epidermis thickening, the imaging quality is gradually degraded because it has to penetrate the thickened epidermis more over time. So that is the one reason the imaging quality is somewhat, a little bit degraded from day 0 to day 2, or even at day 3 and day 4. And then, but by using a two-photon microscope at even longer wavelengths, we can mitigate those imaging quality degradation problem. And then another question is, I would like to ask the atopic dermatitis model. What kind of a model was used for imaging and what are the difficulties? Okay. Yeah, as far as I remember, it was induced by some chemical. So I'm sorry, I forgot the details. But this is a very common, atopic dermatitis model. Uh, and then there was not too much difficulties in imaging this atopic dermatitis model because as you can see here, we can nicely see the nicely distinguish the epidermis and dermis and the space between. So there was not specific or any special difficulties in atopic dermatitis model in analyzing the drug penetration, as you can see in this result. And then, okay.
Yeah. So thank you everyone for all the questions. So as demonstrated in today's presentation, IBM Technology collaborates with a wide range of institutions and research centers to provide CRO services. If you're looking to support your research or are interested in potential partnerships, we encourage you to contact us at any time. This webinar recording will soon be available on our YouTube channel. If you have any additional questions or if we weren't able to address your inquiry during the Q&A session, please feel free to reach out to us at information@ibmtech.com. Thank you again for joining us today, and we look forward to seeing you at our next webinar. Thank you.
Thank you.